So, dear friends, I am Dr. Singh. You know that I am continuing my lecture on pharmacology and toxicology. And previously, I have uploaded you can say, my video lecture, which was on pharmacology and how we go for pharmacology to pharmacokinetic and toxicology to toxicokinetic. Hope that you have enjoyed that video, and I hope that you like and you will share more and more. So now today I will discuss about the very important topic which will be very useful at each and every step for you people, because you should know a very basic about the toxicokinetic. That means toxic, they are not safe, they are lethal, they are harmful. Kinetic means to analyze those substances which produce a toxic effect in our body. So that means whenever we go for a clinical trial, if we are having a new drug and we have synthesized or we have extracted a new drug, it is very important to decide the safe dose of the drug. So that means safety and efficacy and potency, these are and quality of the drug, these are the major points. So dear friends, you can say if nothing is safe, you cannot consume anything. So that means there is a one statement by the Paracelsus that any drug, any substance can be safe or a toxic, only those differentiate. So dear friends, it is important to know that whenever we go for the synthesis of the drug, then we go for the in vitro analysis of the drug, so that we can predict IC50 value of the drug. From that, we can derive the safe dose or a in vivo dose for the animals. So today I am going to discuss with you about the animals, because animal studies which are called as a preclinical studies. So what I can say that before clinicals, that means you know that whenever we do any drug trial, for the trial, whenever we want to file a drug for a clinical trial, they need a preclinical data. That means, what means pre? Pre means before to clinical analysis. This is called as a preclinical studies. So dear friends, we do preclinical studies in case of animals. So many students, they every time ask the question that how we go for the preclinical study, what are the suitable species for the preclinical studies. So in this lecture, I will explore more about these things, so that you should know that whenever we move from the preclinical to clinical phase, there is a requirement of data that need to be submitted for investigation of a new drug in a clinical trial. That means in India, which you will get approval from SEDISCO, or we can say on the other countries, there may be having approval from FDA, which is called as a US FDA, China FDA, Japan FDA, Malaysia FDA. So like in India, we are having a SEDISCO. So that means we need approval to do the trial if we want to think so our drug will be a novel and beneficial for the society. So that means we should select some animals, and these animals which are basically selected are of two types. They are called as a rodents and they are called as a non-rodents. So there is always one question by the student, so why they are called as a rodent? Why they are called as a non-rodents? So that means rodent means they contain a few teeth in their mouth. That means they contain the incisors. In the rat mouth you can see there is a upper two teeth and lower two teeth. That means all the whole of the jaw is not fully occupied by the teeth. It means those type of species which partially contain a teeth on their jaws, they are called as a rodents. So it is very important that rat, mice, these are animals which are called as donkey, it can be pig or some other animals. So if anybody mostly asks question, sir, why we go for rodent to non-rodent? Why there is a need of data of both the species, that is rodent and non-rodent? So basically, dear friends, I will tell you one simple thing, that we are going to keep or to move our track from the preclinical phase to clinical phase. Now in the preclinical phase, most probably all the animals, they are not 100% show matching with our body in terms of behavior, in terms of physiology, in terms of anatomy, in terms of genetic, or in terms of some other functions. So we cannot assume that if we take a data of rodents only, like rat or mice only, the data should be sufficient and that data can be considered for the clinical trial. No. So that's why we should go for the analysis on both of the species, so that we will consider a rat, we will consider a mice, because they show a good behavior changes in their body that are most probably like to human but not 100% match with the humans. Likely we go for the dog. So dog was the preferable animal from the old time for the cardiovascular studies, because dog is having a large heart and dog heart effect can easily be analyzed. Similarly, a cat, which is called as a non-rodent, cat was from the old time used for the learning and memory. And on the other thing, there is some scientific thought that a cat having an age of 1 month contain an equivalent number of neurons in the brain that are equal to the baby or a small children with the age of 1 year. So what can you imagine that 1 month cat neurons in the brain are equal to 1 year baby of human. So that's why the cat is a very safe and you can say a good species which can be used for the memory analysis. That's why monkey, they are preferred for the learning, they are preferred for some other activities, and likely there is a one species which is called as a kinetic, which is called as a non-rodent, which is preferred for analysis of histaminergic response of the drug, because their trachea or the respiratory tract of the guinea pig shows sensitivity similar like a human being. So that's why it means don't go for the blind selection. You should go for the selection of animal on the basis of scientific concept, or you should go for the selection of the animal on the basis of behavior similarities, or you should go for the selection of animals on the basis of some characteristics, anatomical, physiological, phenotypical, which should be matched with the human beings. So dear friends, I will conclude this only, that whenever you go for the species selection for the preclinical trial, both type of the species are selected. What it means? Rodents as well as non-rodents. Now this is the question, why need toxicokinetic studies? That's why we ask sometimes question, why need pharmacokinetic studies? Because after the preclinical trial, whenever we go for the post-marketing surveillance, there are some effect of the drug which has been analyzed to be toxic, or they produce ADR. This is a part of pharmacovigilance. In the separate lecture, I will discuss about that, what pharmacovigilance do, how they generate signal, and what are the essential components for a signal generation. That will be a separate topic I will discuss with you people. But now I will discuss about that why we do need or do toxicokinetic studies. It means simply it is the kinetic data where we exposure and the That means whenever a drug go beyond the safe window, therapeutic window, drug can go for the toxic effect, produce toxic effect. So what it means, we can do safety risk ratio analysis. That it is a very important and essential part in the clinical practice, that whenever we do the, you can say, post-marketing surveillance in the phase 4 of clinical trial, sometimes the drug, they are failed, they are withdrawn because their risks are more and their safety is less. But until and unless we should know that if there is availability of some alternative drug, then we can go for the drug withdrawal or drug ban. Otherwise, we can check the safety risk ratio, that whether the drug are safe or they are toxic. Now another thing, that we check the safe dose level of the drug. I will give you a simple example of the dose response curve, that whenever we do DRC using a organ bar, we always use some of the drug like acetylcholine, their antagonist, and we do some assays. Okay, they are called as biological assays. The basic aim is that we should know the least concentration of the drug which is able to produce a response, or which is responsible for causing a tissue contraction. So this is from the DRC. Now another question which is called as a goal of toxicokinetics. I will discuss with you people from the objectives. What are the objectives? So there should be two objectives. One is called as a primary objective and another is called as a secondary objective. So primary objective means whenever in the animal we want to check the relationship of dose level and time course of the toxicity, because there are 2 to 3 things, that sometimes drug can produce toxic effect immediately, sometimes drug effect can be delayed, sometimes drug can produce a toxic effect at the end points. So that means if you cannot imagine that if we have taken a drug for 5 days, 2 days, 3 days, and we have not found any ADR, you cannot say it is safe, because drug can produce a clear type of ADR. Drug can produce a ADR, sustained type of ADR. So that means these are the problem. Another is the secondary objective. In the secondary objective, you know that you do the study, that the toxicological finding, and you do the assessment of human safety clinically. So that means we will go for the dose titration analysis whenever we see that drug is a toxic. Now then we go for the titration, that which will be the suitable dose that can be considered as a safe for our health. Now other that whenever you do this toxicological finding, so you should know both type of the consequences, that it can be able to produce a toxic effect on the body, it can be able to produce a toxic effect on a one organ, or it can produce a toxic effect on a one cellular or a tissue level. So that's why it is very important to understand these type of the things. Now, the number of questions many times the researcher asks, that what are the different type of toxicokinetic studies which we should perform for a drug. So in this lecture I will discuss about a few studies that we will do for the toxicological profiling. One is called as a acute, another is called as a subchronic, and third is called as a chronic. That means there are 3 phases. This depends upon the drug used, that it can be used for a acute time, subchronic time, or a chronic time. So dear friends, if it will be a acute, it can be acute oral, acute dermal, acute inhalation, or skin sensitization. Now easily you understand that whenever we go for the toxicology analysis for 7 to 28 days and maximum 1 month, this can, you can say, it is a short-term toxicity, or it is an acute or a subacute. So whenever you go for a 7 days, 14 days acute, 28 So that means subchronic, which can be up to 90 days, because this data is required for whenever you want to take your drug for the clinical trial analysis. Then you should know subchronic dermatotoxicity on the skin, or inhalation, or one generation, two generation, because you cannot imagine that one drug can be too safe for the body, on the other side it can produce some toxic effect on the reproductive system. So if it can affect the reproductive system, it can affect the whole of the generations. So that means we do toxicity of one generation on the parents, and we do toxicity on the second generation, their offspring, which is called as a F1. Now, another is called as a neurotoxicity. I will discuss separately on the neurotoxicity, what are the drugs which are neurotoxic, and what are the consequences of the neurotoxicity. So dear friend, this is the last I will discuss with you: chronic toxicity, oral, dermal, inhalation, skin sensitization, which can be for 12 months. So another one called as a long-term toxicity. One is called as a carcinogenicity, another is called as mutagenicity or genotoxicity. So that means there are these few types of the studies which are conducted for the toxicological analysis. In the coming lecture, I will discuss with you about the individual type of toxicity, how you should conduct, what are the